Okay, hi there. This is a fairly brief lecture recording, and this is chapter 10, and we'll look into some of the sensory physiology. And what I'll talk about is some general properties of sensory and of the sensory system, and that's section 10.1. Then I'll talk specifically about somatic senses, 10.2. And then we'll spend a little bit of time on sensories in terms of the special sensory organs. And that's section 10.3. We'll focus on olfactory and gustation. You also have vision in there. You can take a look at it in reference to your lab activity so that you can connect to that and find the source that allows you to work through the lab. But I'll go through the examples of smell and taste. So let's take a look. Some general properties of the sensory system. One, we want to remind ourselves that we're talking about the afferent division, right? The sensory division of the nervous system, in particular, the peripheral nervous system. So that means receptors and sensory neurons that then send the information to the central nervous system. Special senses are, of course, vision, hearing, taste, smell, and equilibrium. Somatic senses are particular to either the skin or stretch receptors associated with your joints. So you have touch, temperature, pain, itch, proprioceptors, so proprioception, stretch, and these are all inputs that are coming from the outside, and that is what the term somatic means, from the skin, basically, so from outside, rather than visceral sensory coming from the inside of the body. Stimulus has a physical energy, and a receptor in your skin, for instance, acts as a transducer to take that stimulus physical energy and transduce this into an electrical potential energy. The intracellular signal then is usually a change in that membrane potential that we know so much about and that we know so well when it comes to potentials. The stimulus, depending on its strength or frequency, creates a change in the membrane potential that reaches threshold. If it does reach threshold, we create an action potential, and that action potential is then traveling through the sensory neuron, in this case a somatic sensory neuron, and makes its way to the spinal cord in the brain. Now, if the stimulus is not strong enough, it'll be a graded potential. And a graded potential is a depolarization that is not to threshold, so that means there might be a more frequency of that stimulus where the graded potential add up and then cause an action potential. The information is then integrated in the central nervous system, ultimately makes its way to the cerebral cortex, and is acted on subconsciously if it, for instance, goes to the limbic system, which it actually does go either way. But there can be, of course, subconscious reactions. As an overview of the sensory processing and the division, take a look at table 10.1, information processing by the sensory division, by the afferent division. It shows you the special sensory as an overview, and it shows you somatic senses as an overview, as I introduced you to. And then the processing, somatic stimuli from outside, visceral stimuli from inside the body. So examples are muscle links and tendons, and that's proprioceptors, that's about stretch. Or, for instance, blood pressure. Those are all examples of visceral stimuli. I want to talk a little bit about the types of neural receptors that there are before we take a look at the table here. As an overview, we have receptors that are either naked, and those are usually free nerve endings, or they're encapsulated. They have connective tissue around the receptors. And then, of course, the associated neurons can be myelinated or unmyelinated. Non-neural receptors are present for certain types of special senses, and they are highly specialized, and they're usually associated with a nerve. So you don't have a receptor just not associated with a neuron. There are some accessory structures that enhance the sensory system. For example, the somatosensory receptors, the receptors that are about your skin and spatial mapping to the brain. We have four major groups that you can see here in table 10.2. You have chemoreceptors that respond to chemicals, whether it's oxygen, pH, or organic molecules, or such as glucose. And we have mechanoreceptors, and mechanoreceptors respond to mechanical energy change and pressure or sounds. So pressure, those are specific kind of mechanoreceptors known as baroreceptors. They play a role, for instance, in blood pressure regulation if it's a visceral system. Cell stretching, osmoreceptors, vibration, acceleration, and sound. All of these are examples of different types of mechanoreceptors. Then, of course, there is photoreceptors. Photoreceptors are about vision, and they respond to light and contrast, so varying degree of photons in reference to the visual light and their particular wavelengths from about 350 to about 750 is the visual light spectrum for us. And then another type of receptor, of course, are thermoreceptors. They're all about temperature. They respond to temperature change. It's cold and hot. And within that area, we have multiple subcategories. So table 10.2 gives you a bit of a brief overview. I do want to talk a little bit about sensory receptors and how they function. You have, just as a simplicity, I just told you we have free nerve endings and we have capsulated with connective tissue, and here are some examples where you have a free nerve ending, a stimulus, and that free nerve endings is kind of like the dendrites, right? So these are the receptors associated with the axon and to the axonal terminal. And here is your cell body of that, let's say, somatic afferent fiber. And the area from the free nerve ending receptor part to the cell body, and from the cell body, most likely to the spinal cord, the axonal terminal and axonal terminals will end in the spinal cord. This is usually unmyelinated or very lightly myelinated. Then you have an example here of a little bit more complex neural receptor, where the nerve endings are encapsulated with layers of connective tissue. The stimulus is received by this receptors. The unipolar somatic afferent fiber is myelinated, as you can see here, the myelination. It's unipolar because we have the cell body here and we have only one projection off the cell body, so they're called unipolar. Very dominant in the peripheral nervous system, the PNS. And then here you have an example of a very specialized type of receptor, sensory receptors, where cells release a neurotransmitter onto the sensory neuron. So here's a synapses to the sensory neuron. Here's the specialized receptor. Upon receiving a stimulus, they'll release a chemical, a neurotransmitter, that then initiates the action potential that travels along the myelinated axon and makes its way to the axonal terminal, where it will release its neurotransmitters and initiate an action potential with the second neuron, until it makes its way up to the central nervous system. So that's kind of a three-way demonstration of the different types of receptors associated with the different types of afferent sensory afferent fibers. Sensory transduction, I mentioned that a stimulus has an energy and a physical energy, and this energy needs to be converted into something that a neuron can work with, and that usually is all about membrane potentials. It's about electrical stimulus. And so the receptor's job basically is to respond to a specific stimulus that it's designed to respond to, and then turn that physical energy into a membrane potential change. So that membrane potential change generally responds to ion channels opening, but it can also be initiated by a second messenger. And that second messenger often is calcium, but it can also be ATP. If the stimulus is adequate enough, that form of the energy is transduced into the receptor energy, and that can be a graded potential, or it can lead to an action potential. The adequate stimulus will then, of course, mount a response of the afferent system. And just like in every other membrane change, in order to initiate an action potential, there is a threshold. And that threshold is a depolarization value at a minimum stimulus. We talked about this in the sense of being a resting membrane potential being at minus 70 millivolts, and then a threshold stimulus would have to reach depolarization to minus 55 millivolts. And that same principle applies to sensory transduction. So the receptor potential, which is initiated by the stimulus and the changing of the membrane potential of the receptor, is generally a graded potential. The change in the sensory receptor membrane potential is exactly what a receptor potential is, and that can lead to an action potential. Now, what we have here is a little bit of a dynamic of how the receptor is located in a particular area and what it responds to, how it responds to, and how it connects to neurons. So the receptor field, by definition, is literally a physical area where a stimulus activates a neuron. So if that's in your skin, that could be this big, or it could be, if it's on your fingers, really tiny, right? So receptor field size varies. The primary sensory neuron and often secondary sensory neurons are associated with the receptor field. So the primary sensory neuron is always that neuron that is associated with the receptor that receives the stimulus. The secondary neuron is the neuron that is communicated to with next. This neuron is potentially the one that then elicits the action potential, and the action potential travels towards the central nervous system. So we have a primary neuron communicating with a secondary neuron. The receptor field frequently overlaps with other receptor fields, neighboring receptor fields that can occur. It doesn't have to be. You see some overlapping here and then very separate here in the illustration, this illustration in figure 10.2. The size determines the sensitivity of that area in your body. In your lab, you may be able to do the 2-point discrimination where you can use this compass if you have one. If not, I think I gave you some ideas on how to create one. And you keep your eyes closed and you have someone help you with this. You need a partner, so maybe in your family member, and they will come down, let's say, on the skin simultaneously with your eyes closed and hit this area simultaneously. And it doesn't have to be hard, very little, doesn't have to be, don't draw blood or anything. Just kind of come down simultaneously. And then you, with your eyes closed, are to evaluate or determine whether that is one point that you're feeling 2 points that you're feeling. So that's what we call the 2-point discrimination. The distance can be measured with a ruler. At what point can you distinguish that it's 2 points rather than 1? And of course, that correlates with what you're seeing in terms of the size of the receptor field. If you have a lot of small receptor fields, you're more likely to distinguish that it's 2 points because it's initiating 2 different sensory neurons. So 2 different receptors. Whereas when there's an overlap, it becomes a little bit unclear and you can't tell, is it really 1 or it's a very large receptor field? Is it 1 point or is it 2 points? Because it also just communicates to 1 neuron. So the size of the receptor field gives you an idea of the sensitivity, right? Less sensitive areas have larger receptor fields. More sensitive areas have very small receptor fields. I mean, you don't have to be the subject. Your friend or family member can be the subject. In terms of the sensory pathway in the brain, which I'm sorry, that should say brain, I'm using figure 10.3. And before I kind of go into that, I just want to tell you a little bit about sensory information. So we have a receptor that stimulates an afferent fiber, and that makes its way to the spinal cord, the action potential, and then from the spinal cord it has to travel to the higher regions of the brain. So the spinal cord to the brain is what we call the ascending pathway, the afferent pathway. You've heard me say that before. And therefore is a direct connection to the brain, and we travel along the spinal cord along the brain stem and then into the diencephalon. Depending on where the sensory stimulus is coming from would then determine whether we're in need of the spinal cord. So some special sensory receptors and organs like the eye or the tongue or hearing and even equilibrium, they actually either go into their afferent fibers transmit either into the brainstem or into the thalamus. So they're not utilizing the brainstem, and that has to do with their location, proximity to the brain. So visceral reflexes are integrated also in the brainstem or along the spinal cord to the brainstem. Usually do not reach conscious perception. So your cerebral cortex is all about your consciousness, usually stay within the autonomic nervous system. Perceptual threshold is a term that's used that allows us to understand that a certain level of stimulus is necessary for a particular sensation to be perceived by your brain. And it's the brain that actually does the perception. So your receptor doesn't. Your receptor is just a membrane potential change. The sensory neuron is just an electrical signal being sent. Until it reaches the brain, there is no perception of that stimulus. So if you think about when you're smelling something or when somebody touches you, how fast the perception occurs. Gives you an idea how fast that information travels from your skin all the way to the conscious perception of your cerebral cortex. Super fast. So perception happens on the brain. Habituation is a term that comes up often with receptors because habituation is a decreased perception of a stimulus. And that usually has to do with the continuation of the stimulus, and there is a inhibitory modulation that has to kick in. So I don't have to be told over and over again about the same sensory. For instance, when you have clothes on your skin. So your receptors habituate. So there is an inhibitory modulation that occurs on that receptor level to prevent the constant perception on the brain level. And often that ties in with the function of certain receptors adapting to stimuli versus others adapting very slowly, and some not adapting at all. There are some sensory that where the receptors cannot adapt to and shouldn't adapt to. Pain is one of them, and proprioceptors of your joints. Those receptors should not be adapting at all. So no habituation should be taking place, because your brain needs to be aware and constantly told what the body's position is in space in order to be able to balance and calculate your moves. So each major division of the brain processes one or more types of sensory information. Again, it depends on which one we're talking about. So most pathways are 99.9% pass through the thalamus, and that's because it is the sensory relay station. And the thalamus edits the sensory information and then is able to send it to the particular location because of the association with neurons that are being stimulated. So it will most certainly be sent to the somatosensory cortex if the stimulus comes from outside, so through your skin. And then the homunculus is the map of all the receptors on your skin, and that can identify where this is happening. But it's also sent to the different cortices that identify that type of sensory. So if it's taste, or if it's smell, or if it's vision, right? Those are the primary cortices we talked about, and the association cortices that allow us to interpret and understand and put meaning to it. We also have information sending to the cerebellum, particularly when it comes to balance and sound. So if you take a look at these little boxes here, olfactory pathway from the nose projects through the olfactory bulb to the olfactory cortex. We'll look at that a little bit closer. Most sensory pathway projects to the thalamus, that's right here, and then gets sent to its particular destination. Equilibrium pathway projects primarily through the cerebellum rather than directly to the thalamus, hence 99.9%. And that has to do with the function of the cerebellum being responsible for equilibrium and balance. This is figure 10.3. Coding and processing. It could be complex, it could be simple, depends on how you look at it. Sensory modalities are those modalities that we just talked about, right? Primary modalities, vision, hearing, sound. And so which sensory neuron is activated determines what area of the brain is being activated. And that labeling is one-to-one, one sensory neuron to one area of the brain. And that is associated to one receptor of that sensation. So it's a one-to-one relation. The location of the stimulus is tied into, of course, the receptor field in terms of the accuracy. So the receptor field is activated due to the stimulus, and that allows the identification as to where that stimulus is happening. We also have lateral inhibition, and lateral inhibition prevents the kind of diffusion of that, that's Fresno, diffusion of that receptor field to a wider location. So inhibiting the population of sensory neurons around that receptor field that's being stimulated prevents it from being kind of unclear. The intensity of the stimulus has to do with the number of receptors that are being activated, and of course the frequency by which the stimulus is activating the receptor. The duration of the stimulus is really the duration of an action potential. It's generated, sent, perceived, respond. Receptors do adapt. I've mentioned that. We have tonic receptors and phasic receptors. Most receptors adapt in terms of continuous stimuli, except for pain, nociceptors, and proprioceptors. Here's an interesting figure, 10.4, that shows you how the brain uses timing differences to localize sound. And it's minimal in terms of the perception for us consciously, but for the brain functions in terms of electrical impulses and membrane potential change, it makes a difference. So when the sound comes from one side, let's say the right side, then the traveling time of that sound to the right ear and then signaling ultimately to the auditory cortex on the right side is much shorter in terms of those sound waves versus hitting the left ear. And that time difference really allows the localization of the sound for the brain to occur so that you know, okay, it's coming from the right side rather than the left side. Kind of fascinating. I've mentioned lateral inhibition. Lateral inhibition enhances the contrast and makes a stimulus easier to perceive. Hold on. That's a breadcrumb for this lecture. If you are watching this lecture, I have you know that neighbors came by with a dog and Fresno saw them and is very upset about it and is barking. If you are listening to this lecture, that's what you're going to email me. All right, so talking about lateral inhibition, to make it a precise and contrasting stimulus experience, I'll just use this example that you see here in figure 10.5. You have a receptor field, and this receptor field is associated with receptors, and we have A, B, and C. And these receptors are then, the primary sensory neurons are then communicating with a secondary sensory neuron and communicating with a tertiary neuron. And that's about the length it takes to get from the skin to the brain. So the tertiary neuron is the last one that actually is sending it from the thalamus to the somatosensory cortex. So this is the first neuron, and you have a pin that is pricking pretty much in the dead center of a receptor field, and that only initiates our primary sensory B. So it would behoove us to have a mechanism in place that would inhibit the sensory neurons that are laterally located to that receptor field in your skin by inhibiting them and only allowing that stimulus to travel from primary sensory B to secondary neuron B to tertiary neuron B, and then allowing the precision of that stimulus and the clarity of exactly where that stimulus occurs. So inhibiting the neighboring neurons when they're not being specifically triggered is due to inhibitory neurotransmitter release, right? Inhibiting neuron A and C. And that creates a nice contrast in terms of the neighboring area and allows your brain to very specifically perceive. So stimulus, primary neuron response is proportional to the strength of the neuron. The pathways closest to the stimulus will be inhibited, so that would be here C and A, and that allows inhibition of the lateral neurons but enhances the perception of the neuron associated with the receptor field, in this case B. Right? You can take a look at the graph. If you measure those perception and membrane changes, then it's B that really has our highest contrast response. Let's take a look at somatic sensory, touch, proprioception, temperature, and nociceptors. Nociceptors are about pain and itch. Temperature, of course. Proprioception is about stretch, and touch is about pressure. Somatic, meaning from the skin. The pathway for somatic perception projection to the cortex in the cerebellum. So receptors in your skin are associated with afferent fibers, and it has to make its way through the somatosensory cortex, but also to the cerebellum. And that's where the perception occurs. Receptors for somatic sensories are in your skin, but we also have receptors in the viscera. A little tricky. They're really visceral receptors. Your primary sensory neurons, as we just saw, synapse in the central nervous system, and that is from the receptor in the skin along the afferent fiber to the spinal cord. That's your primary sensory neuron. That will synapse with a secondary sensory neuron that is within the central nervous system, in this case, the spinal cord. The secondary sensory neuron is often an interneuron within the spinal cord that will synapse with a tertiary neuron. And that secondary neuron can reach all the way to the medulla oblongata, or it can reach all the way to the thalamus. The tertiary neuron is then that neuron that sends from the thalamus to, let's say, the somatosensory cortex. So this is in series, right? This is in series pathway that it travels. So it's not just one neuron that travels from your skin all the way to the brain. It's generally 3 neurons. And unless we're talking about those special ones that are closely associated with your head, they're not going through the spinal cord. They're either going to the thalamus directly or the brainstem. So this is just kind of showing you exactly what we talked about, the somatosensory pathway. You have here the primary somatosensory cortex, which is located on the postcentral gyrus. And you have a stimulus that comes, let's say, from pain or temperature, and it travels from those receptors and having been generating a graded potential, an action potential, traveling along the sensory neuron, and your primary sensory neuron forms a synapse in the spinal cord with your secondary neuron, and then it depends on what the type of sensory is. If it's a fine touch, proprioception, or vibration, then it most likely will synapse and travel to the brain stem. If it is about nociception, pain, and temperature, the interneuron will actually be traveling on the other side. It will change sides on the spinal cord level rather than the medulla oblongata level. And so it will shift over already interneuron from one side to the other side and then travel to the thalamus. So as you know, left hemisphere controls the right side of the body, right hemisphere controls the left side of the body. So at one point or another, there has to be a crossing over. Otherwise, this doesn't make any sense. So when you take a look at touch and vibration, most of those sensory signals are sent along on the same side of the receptor, along the spinal cord, and then they cross over in the brainstem, specifically in the medulla oblongata, where this is now the secondary neuron to the thalamus has already crossed over, and from the thalamus to the somatosensory cortex. Unless it is something like pain and temperature, then the crossing over happens at that location within the spinal cord already. And so your interneuron your secondary neuron is already synapsing on the same side, and then it is structurally crossed over already and makes its way all the way to the thalamus and tertiary to the somatosensory neuron. Now, I just want you to see that. I just want you to understand that there's 3 neurons in place, and that at one point or another, there has to be the crossing, decusation occurring. And it can happen on the spinal cord level, and that's usually with pain and temperature. And for most other sensory, that happens in the medulla oblongata. This is figure 10.8 that tries to illustrate that pathway, that afferent pathway. Here's just the somatosensory cortex, figure 10.9, the homunculus. I've showed you this before. The size of the projected cartoon tells you the size of the innervation of that area. So your face and your hands usually have lots of sensory receptors, so lots of innervation, and that also determines, of course, how sensitive we are. If you take a look at the skin, the receptors in the skin, there's a whole variety, and we have cutaneous mechanoreceptors. You may remember this from anatomy. If not, this is a picture of a skin section. I can post that. Let me know if you want me to post that model where you can see those receptors inside the skin. So cutaneous mechanical receptors, so in the skin, mechanoreceptors are responding to deformation. So we said, for instance, pressure and touch are mechanoreceptors. And some examples in your skin are the Pacinian and the Meissner corpuscle, but also Ruffini corpuscles or organ. And the difference between those receptors is really the depth of the location within the skin. Free nerve endings are penetrating the epidermis, so they're fairly far up. But then if you take a look at the Pacinian or even the Ruffini corpuscles, they're a little bit deeper. They're in the dermis or even in the close to the hypodermis of the skin. And so that means that that mechanical stimulus has to be deep. It has to trigger them this far deep in. So ultimately, it is kind of a neat trick where we just distribute receptors in different depths of the skin, and that helps in the determination of how strong that stimulus is. So if it's a really, really deep pressure that stimulates the Ruffini corpuscle really deep, then that helps on the brain's perception level to identify what kind of stimulus this is. But also, what this plays a role in is habituation or adaptation. So if you put 1 penny on your skin, right, then the mechanoreceptors will adapt within seconds and you won't feel it. But if you put 2 more in, then it reinitiates a deeper receptor because there's more weight of 3 pennies or 4 pennies. So now these will be triggered until they habituate. So most of these mechanoreceptors will adapt, will stop sending a signal. But these are just some examples. We have the free nerve endings that are associated with an afferent fiber by mere receptor being the dendrites of that nerve. We have a Merkel receptor that is part of the epidermis, although the deepest layer responding to pressure and texture change. You have Meissner corpuscle that responds to like a flutter stroking movement, and they're located in the dermis. You have the Pacinian corpuscles that sense vibration. They're a little bit deeper in the dermis, and the Ruffini corpuscles that respond to stretch. And then stretch, of course, is also a deeper type of touch. All of these will communicate to a afferent neuron that sends the signal then to the spinal cord. I hope that this is clear to understand in terms of cutaneous receptors. You have some lab activities that I hope will solidify this information. Referred pain is another very fascinating concept. And referred pain really is about the map of the body. I've seen better pictures, but this is the one that you have in your book, figure 10.11. And your entire body has a map of these somatosensory receptors and receptor fields. And for some reason, we are able to determine internal pain, visceral pain, by pain on the surface of your body, on the skin. And so, for instance, if you have pain in this area here or on top of your right shoulder, then there might be a liver or gallbladder problem. Whereas if you have problems with your heart, you know this, right? Then it's that left chest and down the anterior aspect of your arm is pain. Often also the cheek, actually. So pain in those superficial area of the skin are indicating pain that is inside or a problem that is inside. And so we consider that a referred pain. There are some theories about how referred pain works. There's lots of information out there. McGraw-Hill is one source that your book uses. It's a different author. But one theory that I think is probably the closest one is that nerves are packaged up with multiple neurons. And a somatosensory neuron can be packaged up with a visceral sensory neuron. And that stimulation of a sensory on the skin that is associated with a neuron that is, for instance, associated with the kidney, that proximity can cause the stimulation, unmyelinated axons, the stimulation of that visceral sensory neuron, and is simultaneously sent. So if there is an issue with the kidney and it's affecting the sensory neuron, then the proximity to that somatosensory neuron causes a stimulation, and then that information is traveled along the ascending pathway and identifies as, oh, I have some pain right here. And so when you communicate that to a doctor, then they're like, this might be a kidney problem. So kidneys is often in the back. Not always, but a lot of the times it's back problems. That's what we call referred pain. This is one of the theories, that you have one nociceptor from several locations converging on a single ascending track and then kind of overlapping. It's not 100% clear how it works, but I think this is a pretty good theory, if you ask me. Let's take a look at olfactory system. We have a few more slides left that I want to share with you. This one and the second one, focusing in olfactory on smell. A shorter pathway, ascending pathway, due to the location of the sensory neuron. And you have an illustration of that here. Figure 10.13, you have the olfactory bulb, which is associated with the olfactory tract. And of course, that's cranial nerve number 1. And the receptors are located in the membrane, the epithelial membrane, and your neurons that are associated are kind of dangling into that. And as molecules come in, causing the receptor potential to create a change in membrane potential, and then that travels along the tract, the olfactory tract, to the olfactory cortex, and from there goes to two locations: your somatosensory cortex, sensory cortex, and the limbic system, the emotional brain. Smell is really tightly connected into forming memories and storing information. So the limbic system is incorporated. Chemoreceptors are those receptors that respond to smell, but also taste, and that's what we're looking at here as an example, the olfactory. Olfaction is one of the oldest I don't know how that works. I think oldest understood senses. It allows us to discriminate amongst millions of different odors. Some are better at it than others. The olfactory bulb is an extension of the forebrain. The olfactory pathway is the olfactory sensory neuron, which is cranial nerve number 1, ascending pathway to the limbic system, specifically to the amygdala and the hypothalamus, and also linking to the somatosensory cortex. So smell is really linked with memory and emotion. I hope this kind of makes sense to you. Let me show you the other chemoreceptor, taste, and that's kind of cool. So you have your tongue and you have these taste buds, and there's a pore, chemicals bind to it, different kinds of sweetness, umami, bitter, and sour. And you have these receptor cells, different types of receptor cells, type 1 and type 2. And they respond to the chemicals. And depending on what kind of modality is, or submodality, so your sensory is the modality taste, the submodality then would be sweet or bitter or sour, determines what the receptor will do. Will the receptor release a neurotransmitter like serotonin to initiate the potential change in the sensory neuron, or will it release ATP? ATP is a novel messenger, so ATP can elicit a membrane change in the sensory neuron that then travels its pathway to the gustatory, taste is gustatory cortex. So gustatory is taste. Again, taste and smell is closely connected. There's 5 basic sensations in terms of taste. I've mentioned sweet, bitter, and umami is savory, so spicy tastes, and they cause the release of ATP from the receptor type 2, and that activates gustation, that activates the afferent fibers. And sour taste causes the release of neurotransmitters, like for instance serotonin. Type 3, presynaptic cells, salt taste is initiated. Some non-traditional taste sensation would be hunger and salt appetite that then cause the sensory information to go to the hypothalamus to initiate hunger and thirst and for you to respond to those. That's really all I wanted to share with you in terms of sensory receptors, and I'm hoping that it works well with your lab that you will conduct soon, and finishing this lecture on receptors and afferent division, chapter 10.